السلام عليكم. بدنا نبلش محاضرة اليوم. The lecture or the seminar or whatever it is. It is about DNA purification and detection. It is quite possibly the integral lecture or seminar for this course. راح تشوفوا إنه the content of هاي المحاضرة هو أغلب إشي راح تشوفوه غالبا بكل العالم. Wherever you go. The contents of this lecture include PCR, gel purification, and DNA extraction. Is probably the most common, or the most common or the dominant molecular diagnostic technique in molecular biology. It is also the technique used in a lot of things. لا أشوف ال PCR له كتير استخدامات مش بس بالdiagnostics. You'll see in cloning, in research, in detection mutation, in gene expression. There's a lot of techniques and a lot of applications that you can use PCR with. فالمحاضرة كتير مهمة. This is probably this is the integral lecture that you need to give all. في آخرتها راح تشوفوا كمان فيديوهات. I want you to watch these videos. هدول الفيديوهات كتير ساعدونا لما أنا كنت طالب ماجستير فيما سبق. أو فيديوهات مشابهة لهم. يعني it's very important to look at these videos and to see يعني it's just a good visualization for what we're going through today. فاللي أنا يعني المهم بالكورس كله شرحنا إلى الآن عشان نقدر نشرح اللي بنحكيه هلا حاليا. So we are getting an input that we're looking for اللي هو DNA template that we're going to test واللي هو عادة تحصل عليها من DNA extraction. So the template can be bacterial DNA, human DNA, viral DNA مثل تحصل عليها من human DNA أو من human cells مثلا إذا كان infected cells. So you have multiple types of cells from eukaryotes to prokaryotes that you need to use for DNA extraction. This is the template. هذا هو زي ما حكينا زي ما template strand مثلا. So this is what you're looking for, what you're looking to find a specific DNA sequence in. So to find the sequence that you're looking for, so the template that you're looking for, what you're looking is what you're designing your primers to look for. إذا تذكروا حكينا كلمة primer is used to annotate the segment you're looking for. إذا تذكروا حكينا إنه the DNA polymerase cannot start replicating a specific sequence of DNA unless it has the seat. الكرسي تبعها مش زي ال RNA polymerase. The DNA polymerase it has to have both sequences on both strands. So it has to have a double strand. So what you do is you put a small number of sequences على بداية الجين أنت بتدور عليه أو segment من ال DNA sequence أنت بتدور عليها. And then it's a complementary strand 15 20 25 base pair which you design to be the same sequence at the beginning of the sequence that you're looking for. وتحط primer على نهاية السيكونس بحيث إنه you are annotating زي كأنك you are telling the polymerase to work from here and stop from here, work from here and stop from here, work from here and stop from here. So it will keep generating the same sequence of DNA that you're looking for. إذا كان sequence موجود. إذا كان sequence مش موجود, the primary will not bind, and then you will not find what you're looking for. So it will be negative. So the technique then is PCR. Just سمعتوا عنهم is polymerase chain reaction. So you use this specific polymerase that you use in a specific manner that will enable you to keep copying the DNA over and over. And then you have to have certain methods to see هل ال DNA اللي أنا بدور عليها موجودة? Is it there or is it not there? So the way it is that you know if it's there or not is either by visualization, so directly, or by مثلا fluorescent or antibody methods or whatever. So that's basically هاي المحاضرة. This is what we know. This is what we're going to go through. هلا إذا أنا بدي أبلش بالDNA, I want to start extracting the DNA. So I have a cell, I have a eukaryotic cell, I have prokaryotic cell. I need to start extracting the DNA. You will find that extraction of DNA عادة is takes a few steps. أول step وأهم step quite probably is to break the cells apart. فإذا حكينا إن الخلية عبارة عن a bag, lipid membrane, right? And it has all this cytoplasm inside, and then the cytoplasm contains all these proteins, enzymes, and then the DNA. So you need to have a mechanism to disrupt these membranes, the cytoplasmic membrane or the cell membrane and the nuclear membrane. So you need to break this and then release the DNA. So the more cells you break apart, the more nuclei that you break, the more DNA that is released, and obviously that will increase your DNA yield. So مثلا إذا كنت chemically disrupting the cells, so proper mixing will increase your DNA yield. إذا تعمل mechanical disruption, more fine grinding of your cells will give you more DNA. Heat disruption, the longer and the higher the heat, the more cells that you break apart and the more the yield you بتاخد. هذا من one side of the spectrum. The other side of the spectrum, the harsher the method that you're using, the more likely you probably might shear the DNA that you're using, which يعني في إلها limit معينة. بس عادة these methods are created بحيث إنه to limit the shearing on the DNA and then to increase the otherwise anything else that you want to break. فشو أنا بدي أعمله؟ بدي أكسر الخلية and I want to digest everything else بالخلية ما عدا ال DNA. What do I have in the cell? I have membrane, I have RNA, I have proteins, and I have DNA. So I will use things to digest everything apart from and make sure that I do not digest my DNA. So how do I disrupt? So مثلا mechanical disruption by you can grind the cells. Chemical disruption, you can use chemical detergents زي عبارة عن زي detergent تستخدمه بالتنظيف. Basically you can use that for مثلا Triton X whatever. You can use that to lyse cells. It might be too harsh. Sometimes we use enzymes, protease K, STS, whatever, to break apart these membranes and remove them and freeze the DNA. Sometimes إذا أنت مثلا you just want to break apart cells and you want to do a quick PCR. Sometimes you can do that. This is مثلا a colony PCR. You can just heat the cells, take a little bit sample because in the small copy, and then you can do a PCR and find it will not be very clean, but it will work. So these are the methods where you can lyse your cells. مثلا أطلع هون sometimes an surfactant is added to remove the lipid membrane. The membrane lipids are the one thing. TE buffer buffer can also stop DNA's enzyme. هاي نقطة مهمة from degrading DNA by chelating the cations that it needs مثل magnesium. RNase is added, proteinase K is added to digest the proteins and the RNA. So I digested protein, I digested RNA, I digested membrane lipids, and I prevented DNA from being degraded by DNAs. So that's my goal from lysis. الهدف الثاني is to precipitate the DNA. صار عندي now DNA is there, it's unbroken, it's free, and it is mixed with all the debris, كل الدبري من هاي digested proteins, RNA, and lipids. So my aim now is to precipitate أرسب DNA and remove all this debris. So it is the common أو بدناش نحكي common هلا أغلب الميثودز صاروا by columns. لكن هاي الميثود هي الأورجانيك ميثود البيسك ميثود. كل الميثودز اللي بعديهم بيجي عبارة عن it's basically a step up from or an improvement on this method. فشو بنعمل؟ This is why we add phenol chloroform. This is why it's called the phenol chloroform DNA extraction. Once you add phenol chloroform, that denatures and separates the rest of the proteins and cellular debris from the DNA. It causes lipids and other debris to fall into an organic phase, so you will separate following centrifugation مثلا. You will separate. So you broke up your cells. You have the cell lysate. You add the phenol chloroform. You might centrifugate مثلا or incubate, and then leave it, and then it will separate into two phases. You will have an aqueous phase on top, and then you will have a organic phase on the bottom. كل الدبري بينزل لتحت, and all the DNA will remain solved inside the water in the aqueous phase. So denatured proteins from a middle layer form a middle layer. You might centrifugate, and then the aqueous layer containing DNA is then transferred to a fresh tube. With a pipette, you remove all this aqueous layer and you put it in a fresh tube, and then you have all your DNA, and you have removed all other debris. So the aqueous layer containing DNA is transferred to a fresh tube using alcohol مثلا, to precipitate it using alcohol, which creates a small pellet. فهنا مثلا هاي صورة shows the aqueous layer from the organic layer. في كتير كتات بيستخدموا هاي الطريقة. مثلا بالبروكاريوتيك فيها يعني راح اشوف عليها فيها اختلافات بسيطة من هون. في كت كنت استخدمناها مرة. It will clump all the proteins down, and you just كنا بس نسحب البروتين كله, and then you will have the DNA fresh. So there are small variations, but basically that's what we're looking at. Once you finish precipitating the DNA and centrifuging, طلع هذا البلت الصغيرة. This is what DNA would look like. A tiny small pellet. أحيانا it might not be visible, فدير بالك. And أحيانا it might be loosely stuck to the tube. فأنت بتروح بتقلب التيوب عشان تنشفه, and the DNA might fall. Even if DNA falls, there will be small remnants of DNA. You can still at least do a PCR. ما راح تروح كلها, مش كلها بتختفي DNA, because DNA is a very strong contaminant. You will find مثلا إذا بدك تعمل PCR, and PCR will pick up even one template, even one strand of DNA will pick it up and then amplify it. So that's why you might find it can contaminate this, يعني easily contaminate tubes. So but the pellet usually is strong enough, and you can do the second step, which is purification. هلا سحبت ال DNA أنت, you washed it, and then this is what you need to do. You are trying to wash the DNA now and purify it from two things: the remainders of cellular debris لسه ملزقة بال DNA. You want to remove that, and then you want to remove salts from your DNA, because salts can ruin your PCR. إذا كان DNA تبعتك مش نظيفة, and you are trying to do cloning, you are trying to do مثلا PCR, gene extract, gene expression. Sometimes you will find PCR might fail, or DNA might have enough contaminants that it might prevent your PCR from working. So purification is very important. Purification is basically you take this pellet اللي هي هاي اللي هون, you remove all this water, or all this this fluid. You put 70% chilled ice cold ethanol 3 times. You wash the pellet again, you pellet it, you wash it again, you pellet it, you wash it again, you pellet it, and then you let it air dry. And that's basically how you wash your pellet, and you remove all these extra salt and cellular debris that may still remain on your DNA. So then it's left to dry, air dry, and then that's point your DNA بتصير هيك بلت لحالها. So and then you reconstitute this DNA in PCR grade water. بتدوب هاي البلت في مية. It's PCR grade water. PCR grade water means it's water that has been tested and cleaned ultra pure to not have any DNA contaminants or DNAs that might, or any other salts that might ruin your PCR. So but you constitute it, بتحلها أو بتدوبها in this grade water You then at this point you might go see how much concentration of DNA you have, and this will be your DNA, your raw DNA that you can use to dilute and then use for other things. هلا راح نشوف إنه في there are other methods to purify our DNA and extract our DNA. The organic extraction هو هذا حكينا عنه. You can use this for everything, all sample types. So بيحكي sample like cell, tissue, blood, plants, but really you can use it for everything. But the purity, is the purity high? I agree. أنا هاي the next few slides are from وكتير من هون بروتوكولات مأخوذة من شركات عالمية كبيرة. ف they might push things شوية. بس is the purity high on organic extraction? It's not. It can be high if your technique is good. But really راح نحكي عن two other methods. Column method and magnetic beads have higher purity of DNA. The throughput راح يكون أقل ممكن. بس it's easily scalable. Reproducibility راح تكون أعلى طبعا in the newer methods, just because the older method might have user errors. So what else do we need from this? chemistry. هلا بنحكي عن chemistry لما نمشي على النقاط الثانية. Spin columns بسموهم spin columns or column based methods. Basically, so this is the most common methods اللي هاي حاليا تستخدم. Magnetic beads are now on the rise. كتير تستخدم كمان الآن. Spin columns basically تحط preparation تبعك. You lyse with certain buffers that they provide for you or you can make yourself. بتحطهم بهالكولم يكون زي هيك زي بيشبه النيتروسيلولوز اللي بيكون it's mesh but it's really made out of silica. There's a lot of silica in it, and then you put هذا your preparation and you pre-treated, you lysed and everything, and then you wash it through. You give it time to incubate it a little bit and let it bind, and then you put like wash buffer, and then all the cellular debris, all the salts will go down, and all the DNA will bind to silica. بيحكوا لك there are a lot of theories on why DNA binds to silica. اللي بيحكي يعني في كتير أشياء theories. The only thing that you might really find is that this is through hydrogen bonding interaction with an vitalized or derivatized hydrophilic matrix provided by silica. Really, we don't know. We really don't know until now كله. But we just know it. DNA binds very, very strongly to the silica for some reason. بحكي لك this underivatized hydrophilic matrix provided by the silica and whatever. But تفتح papers ثانية راح تلاقي other explanations. So the DNA binds to the silica, these silica columns. بتحط wash it, incubate it, wash, incubate, and then spin, wash, incubate, spin, wash, incubate. This is why it's called the spin column. Until you purified your DNA. إذا حطيت مية, ultra pure water on the column, the DNA will elute, تنزل. It will unbind from the silica and it will come down, and that's how you get your column to unbind its DNA. كل DNA تنزل, and then you take this and then you do the same thing for them. Can you reuse the column? No. إلا إذا أنت بتستخدمها على نفس البيشنت أو نفس المريض, نفس البكتيريا, نفس السترين. It might. Will it bind the same capacity? Might. But هم يعني it will be contaminated with the older DNA, and they will have a lot of the cell contaminants here. So it's not really designed for that. الجديد, what's really now being used more and more, what I really like is the magnetic beads. So magnetic separation uses two main components. You will find إنه إلها two main components. الأول, you have to provide a magnetic field. لازم يكون عندك راك فياته magnetism تشغله and then it provide a magnetic field. And then in your sample you add these provided that هم بيعطوك إياهم that you buy these magnetic beads, these micrometer sized paramagnetic particles. هلا حطيتهم بالسسبنشن تبعك هالخلايا المكسرة raw whatever you lysed, and then you put these beads which can be selective. هذا بأول homework راح تشوفوها, so you can go through it. So the selective beads are added to a solution which then capture your molecule that you're looking for. The default form or the first form is the iron-based ferrous form of these beads that binds DNA, any DNA. So you can use it to فتمسك هالمغناطيسات الصغيرة تمسك بالDNA, and then you put your magnetic field and you capture only the beads, and then you lift the beads up from your solution and you leave everything else in the solution down. So you can isolate whatever you want. You can modify this application to RNA, proteins, and whatever, and lift them from your crude sample. The processing أقل, there's less need for mechanical separation and very little stress when shearing forces that you use on your target molecule. Very easily automated. هلا في أجهزة كتير راح تيجي it uses these magnetic beads. It can be expensive. هلا أي تكنولوجيا زي ما بتعرفوا أي تكنولوجيا ببدايتها بتكون جديدة بتكون expensive, and then it starts to become, the more you use it the cheaper it becomes. آخر مرة اشتريت الكت I think it was 500 columns for 500 دينار. So it's not that expensive I think. يعني يجوز يعني double the price for the old method, but it has its merits إنك تستخدمها أحيانا. كل ما كان التكنولوجي أقل اعتمادا على اليوزر كل ما كانت less user dependent. فهذه less user dependent. هي الميثود. يعني بتحط هاي الدي ان اي والبيز الاحمر، بتحط هدول الدوائر السود اللي هم the magnetic beads. They will come and bind the DNA. بتحط المagnetic field تبعك and then بتسحبه لفوق، تسحب هذا and then everything else you can أو بتخليه، ممكن تحط الفيلد تخليه بالتيوب and then you decant everything else, you throw away everything else. يا هيك. It depends. Once you incubate, you do that, and then you wash the DNA 3 times, and then you elute, and then you have your purified DNA. There's في شو اسمه magnetic beads / columns. يعني beads جوا column, so it's like a hybrid of the two, which you still need to put a magnetic field on the column to work. هاي نقطة كتير مهمة. هذا اللي أتوقع في منه بالبلد هلا أيام. So these are the methods. هذا الautomation. So الautomation ممكن تحط مثلا 96 wells 128 wells whatever. كل well بتحط فيها نوع من DNA أو sample with lysis buffer which breaks it. بدخل الجهاز بحط this magnetic rod is lowered. It will capture all the DNA with the magnetic beads and then it will lift it up. It will lift it up and then the beads are then released. هذا المagnetic field بنشال in a new fresh well. بنزل لك إياه in a new elution buffer على طول. بنزل لك إياه بسحبه with the magnetic field enabled to بياخد لك ال DNA مع البيدز in a new well. بسحب الرود اللي فيها المagnetic field وبنزل لك الماتيريال كلها in a new well. So that's the automation part. That's what the new machines do basically. هلا هذا كله بدخلنا على البي سي ار. What is PCR? So now you have your DNA template. You extracted it. You are trying to basically polymerase chain reaction. You're doing chain reaction, so you are doing the same reaction over and over and over. Each time, say chain in a chain. Each time, you are doubling the amount of DNA that you have. So in the first cycle, بكل cycle إحنا عادة at the beginning you have a step where is initial denaturation مثلا, you incubate and then you start these 3 steps over and over and over and then you give it sometimes مثلا an extra step to final extension to keep going, and then you cool it down. بس ال PCR هو بيتكون من هدول. basically this is the chain, this is the repeated cycle, this is the cycle اللي بنسمعه بال PCR. The cycle is 3 steps. First, you are increasing the temperature very very high to denature the DNA. DNA would denature. شو بصير يعني DNA denature? The 2 strands will break apart. So this is denaturing. The 2 strands they broke apart. بتنزل temperature شوية، مش شوية، بتنزل temperature كتير. That allows مثلا ل 55، مثلا هون كانت تكون 95، بتنزل مثلا you have to calculate the primer binding temperature. What is the temperature that your primers that you design, that you're trying to annotate the PCR, what is the temperature that is best for them? ممكن انت this might you calculate yourself. بورجيكم ان شاء الله بآخر محاضرة how you can do it easily. في كتير online places where you can design your primers and then software, and وبحكي لك قديش temperature melting temperature تبع البرايمرز تبعينك. So you take it to that temperature. You can إذا كان نوع PCR تستخدم مثلا باللاب، أنت دائما تستخدم the same primers, and you're always using the same primers, and you're always using the same polymerase. مثلا رح تشوف إيش وين هذا بتشوفه بالمستشفى، مثلا فحص لكورونا، فحص ل whatever. It's going to be for the same primers all the time that looking for the same specific sequence, مثلا the hook protein for the corona or whatever. So the same sequence all the time, the same primers all the time. So you can do the melting temperature تعرفها أنت لحالك بالويب سايت ولا بعدين you can you can then try to fine tune your temperature. ممكن مثلا ترفع شوية، تنقصها شوية، ترفع شوية، 5 temperatures فوق، 5 temperatures تحت, and then you can do multiple PCRs, 10 PCRs, and then you can find where you have got the best yield, and then you can use the temperature that temperature all the time. So once you anneal, your primers have annealed, and then ترفع درجة الحرارة مرة تانية لمثلا 70 72 whatever is the optimal temperature for the polymerase to start working, and this is where extension happens. So this is replication will happen again and again. I will make 2 brand new strands من هالدي ان اي. هالدي ان اي تبعتك ممكن يكون جينوميك دي ان اي، راح تكون مثلا من اخر الدنيا لاخر الدنيا. What you are looking for is this part only. That's the specific part we're looking for, and this is where the primers are going to bind. هلا السايكل الثانية you are denaturing this that you just created and denaturing this that you just created. You anneal both of them with fresh primers, and then you extend all of them, and then you'll have 4. والسايكل وراه you denature all 4, and then you extend all 4 بس عندك 8 وهكذا وهكذا وهكذا. That's basically what PCR is. هاي صورة حلوة مثلا. This is how you denaturing, annealing of the primers, and then extension. You're making brand new, and all these هدول نقط هدول all these are DNTPs that you can use in the reaction. شو اللي خلانا البوليميريز شو هو البروتين اللي خلى ال PCR possible? It's called the Taq polymerase. Taq polymerase is the thermostable polymerase enzyme. هل أنت إذا أخذت polymerase 3 من بكتيريا معينة مثلا من E. coli أو polymerase whatever from eukaryotic cells, will it work in a PCR? الجواب لا. It will not work in a PCR because it's not thermostable. إيش معناها؟ معناها إنه the polymerase that we have works at 37 degrees. So it will denature at 95. It will denature at 72. Its extension is also at 35, at 37. فأنت ما بتقدر يعني you can't really do PCR with it. What really revolutionized this whole thing is that we found Taq polymerase, which is a polymerase from a thermophilic bacteria بتعيش بدرجة حرارة عالية كتير. Its proteins are built to not denature in high temperature. هذا بخليهم thermophiles. Acidophile, they do not denature in acid. halophile, they do not denature in salt وهكذا. So if your proteins do not denature in a certain extreme environment, you are a lover for that extreme environment. So the recombinant form can then withstand repeated heatings with no loss of activity. ايش يعني؟ يعني كل cycle ما راح يخسر من activity. We have modified these polymerases to do that by recombination in other bacteria. فهذا polymerase, it works 5' to 3' with 5' to 3' exonuclease activity. What is a standard PCR? What does a standard PCR have? عادة تشتغل أنت بال50. أنا كنت أشتغل 25 مايكروليتر. أغلب الناس بيشتغلوا 25 مايكروليتر عشان يوفروا material. But basically, these are the components of a standard PCR reaction. What do you have? The main things شو هم راح يكونوا؟ Your template DNA. So you are providing the DNA that you're looking for. DNA تبعت المريض اللي عملت لها أنت extraction هلا أخذت منها. You take a specific concentration. عشان هيك لازم تفحص ال concentration تبعها. A specific concentration. بالبكتيريا بجوز أقل شوية، هاي مش أقل أهمية، بس أحيانا إن يعني مش إنه you do not want to overwhelm your reaction. You can do up to a certain point of DNA. So 10 nanogram مثلا of DNA, 10, 20, it might be fine. Then what you're going to do, you want to annotate البداية والنهاية. So forward and reverse primers, that's what you're using. And then you're going to put polymerase. So that's what you have. Your DNA template, your primers, and then a polymerase that will work. تمام. هلا polymerase بده يشتغل شو بده يستخدم النيوكليوتايدز. You have to provide the nucleotide. So that's the deoxynucleotide mix, DNTPs. بتضيف لهم انت كلهم. In sequencing كانوا يستخدموا DNTP mix that is مثلا radio labeled, so you can بالسانجر sequencing, so you can مثلا تقرأ sequencing تبع DNA. So DNTPs are provided. عادة we use magnesium chloride buffer that improves the concentrations, the salt concentration, improves the yield of PCR. You use the PCR buffer, which also has other salts that are specifically made to work for the polymerase that you buy, and you make it to a final concentration to 25 or 50 with PCR grade water, and then you put it in the machine, which you have the cycle for going for you, and then it will create a cycle. That's the standard PCR reaction. Typical cycling parameters زي ما حكينا, you denature عادة at 95 94 حسب البوليمريز اللي بتشتريه for 1 minute. عادة it's not really 1 minute. The time that you really need is depending on the genomic DNA that you're working with, is plasmid DNA, whatever you want. So the larger the DNA that you have, the more time you want to give it to denature. But usually 1 minute is more than enough. Annealing temperature حكيناها, so it depends on your primer. حكيناها نعطيها. So you give it some time to يعني you can give it up to حتى 30 ثانية. It will work. 30 seconds will be fine. But the more you give it, the more annealing you have. The temperature هنا إذا نقصتها عن التمبريتشر ال optimal temperature for annealing, كل ما نقصتها كل ما كان البرايمر binding less specific. معناته it might bind to areas on the DNA that are not the area that you're looking for. معناته ممكن you might get more likely DNA that you do not want. You might get false positive. إذا رفعت الانيلينج temperature راح يصير معك العكس. Specificity راح يصير كتير عالية, and you might get false negative. إمتى أنا ممكن بهمني أرفع temperature؟ إذا I'm looking مثلا for a single nucleotide error, حطيت البرايمر وبديش إياها البرايمر to bind إذا في one nucleotide غلط, فبرفع temperature أنا. إذا أنا مثلا I'm not very sure about my DNA, I have a problem, or I have more sure about what I'm looking for, مثلا ممكن أقص temperature شوية. So that's what you can do here. This is basically this is where user expertise can come into play here اكثر شيء. بعدين extension بيحكي حط لك هون 3 minutes 3D حسب اللينث تبع الجين او whatever you are trying to. عادة 1 minute per kilobyte of يعني كل 1000 نيوكليوتايد اعطيها 1 minute بالغالب is enough. ف 3 minutes for 3 KB is more enough. بعدين هلا بعد ما تعمل البي سي ار تبعتك بدك تشوف ال DNA so you can see is it positive or negative by مثلا ethidium bromide or هل نحكي عنهم كيف or by مثلا real time methods. QPCR هون ندخل لل QPCR. ال PCR حكينا هذا these temperatures you will still use this is what you're using, the polymerase you are using, everything is the same except على standard هنا PCR reaction there is an addition to the buffer. What is this the addition? It is the dye. It is a fluorescent dye that is present in the mix. This fluorescent dye binds to the DNA when it is being built. So the more DNA you build, the more dye gets incorporated into the DNA, and what it is incorporated in the DNA, it emits fluorescence. معناته كل ما زاد تركيز ال DNA أول شيء كان عندي 1 strand صاروا 2 strands I have مثلا arbitrary نحكي 6 fluorescent binding and then 6 fluorescent emission. السايكل الثاني صار 2 صاروا 4 بدل 6 صار عندي 12 بعدين 12 بصير 24 بعدين 4 وهكذا. So it's linear, it keeps going up to a certain point then you start detecting this fluorescence إنه مثلا إنه هذا positive. That's qPCR. That's Q or quantitative PCR or real-time PCR. إيش يعني real-time? Why do we call it real-time? Because you get your results in real time. You see this in real time. How do we see it in real time? هلا بورجيكم. So that's basically what it is. It's the use of fluorescent dye in the reaction, and then you can, without having to do any other detection methods, you will see the detection by fluorescence right away. So real-time PCR or quantitative PCR, it is developed as a precise, efficient, and rapid method for nucleic acid detection and طبعا إيش polymerase chain reaction. So you are multiplying and detecting DNA at the same time. It is based on the traditional method حكينا. However, real-time PCR combines amplification and detection in one step. That's what we just mentioned. It can use smaller amount of starting material than traditional PCR. It is more sensitive. It is possible to quantify the product based on fluorescent detection. هذا مرجيكم إياها كيف. So you can quantify the DNA that you have in the template. تقدر تعرف إنه هنا راح يكون عندك التمبلت مش معروفة. التمبلت استخدمتها بالانتديشن بي سي ار انت already حكيت مش حكينا بناخذها and we detect how much DNA we have. In this sample أخذتها من مثلا buccal cheek for a patient مش بعرفني how much DNA I have. Or مثلا in a criminal investigation or something, you don't know how much DNA is there, so sometimes you want to quantify this DNA. So it's possible to quantify the product based on fluorescent detection, and there is no طبعا فيش processing بعد ما تخلص الapplication. It will give you the detection right away. راح تشوفوا بالمختبرات إن شاء الله بتشتغلوا أو بتشوفوا هذا. There are basically two important outputs that you are looking for in a qPCR. الأول هو the threshold line والأهم الثاني اللي هو the CQ value. So we will repeat this in the coming slides as well, but the threshold line basically is the level of detection or the point at which the reaction reaches fluorescent intensity above background levels. This is the clincher word. إيش يعني؟ يعني هو اللاين اللي أنت بتعتبر فياته fluorescent, whatever fluorescent you detect is just background noise. It's nothing. You already have dye. Dye might bind to template DNA موجود أنت حطيتها. Whatever, it can cause emission. So these are background noise levels. This is not true detection levels. أنت بحطها threshold line. Below it, anything below it that you can see below it is negative. So before PCR, أنت أو software will set this threshold level according to the buffer that you have, according to the dye, according to the software, according to the PCR machine that you have, everything. So basically هو بس خط أنت بتحطه على الجراف and represents إنه أي level above هذا الخط هو positive، أي شيء تحتيه هو negative. So الثانية إيش المهم الثاني هو the CQ value. The CQ value or the cycle quantification value. CQ is the PCR cycle number. هلا السايكل قديش سايكل احنا بنحط البي سي ار عادة من 25 ل 40. The most people that you use 35 ل 40. The most common number is 35. 35 cycle is what people use. هلا which cycle did you start rising above the threshold line? That's the CQ value. امتى بلشت اي سايكل انت بلشت تصير positive. That's the CQ value. So on the cycle quantification value is the PC. خليني أوريكم صورة على السريع. So that's the baseline مثلا, that's the threshold level. You start the PCR. This is basically what the PCR qPCR بعطيك. أنت بتطلع على شاشة وبكون في fluorescence detection. ما في شيء بضلوا هيك ماشي ما في شيء. مرة واحدة بيطلع. So this is the cycle. هون بحط لك number of cycles. مثلا نابل cycle 18. The CQ value have risen above the background level. خلاص this is the threshold level. خلاص طلع. This is the cycle. This is the CQ value. And then the sample will start to rise and then it will plateau. هلا نحكي لكم ليش. So that's qPCR. This is the output that you get from qPCR. So this value tells you how many cycles it took to detect a real signal from your sample. This is important. Real-time PCR runs will have a reaction curve for each sample in the بتستخدمها and therefore many CQ values. So your cycler software calculates and charts the CQ value for each of your sample that you use. هلا ليش البي سي ار بيطلع من هون وبنزل وهيك when it comes to the positive عفوا. The baseline is the initial concentration of the template is too low. So that's why it remains at baseline. So the fluorescence intensity is too low to be detected and only the background signal is evidence. هذا هو البيسلاين يكون تحت البيسلاين. بعدين بلش يصير exponential growth بلش يزيد. After the target yield has reached the detection threshold اللي هو زي ما قلنا تحت. So the course can be followed through the exponential phase اللي هو this is the exponential phase trying to it starts to grow right here. Linear at the concentration increases the available DNA polymerase concentration بصير قليل بقل. So we don't have enough polymerase to do all the new you have a lot of DNA at this point, so the growth will become linear. And then there is insufficient enzyme to continue amplification بصير له plateauing here. So you cannot increase the intensity but تقدر تعمل أكثر من هيك DNA. عادة لما توصل هذه this is at the end of your cycle, so you're not making more DNA, so you're not increasing intensity whatsoever. So the amplification plot, what we really want to see here is this. So the number of cycle. So كل ما زدت template DNA كل ما أنا I need more primers. كل ما أنا ركبت more polymerases. كل ما أنا ركبت شو more dye and made new fresh DNA, more new double stranded DNA. أنا ركبت more dye على السامبل. فمعناته you will have much earlier detection on a smaller on a larger input اللي هو مثلا 1 microgram of DNA. كل ما نقصت ال concentration of the template كل ما بدك تبلش من الصفر أقل. مثلا إذا كان عندك 1 template of DNA عشان الوحدة تصير 2 و2 تصير 4 وال4 تصير 8 و8 تصير 16 وال16 تصير 32. So it requires كل مرة it requires a cycle. So you might not get above threshold level ever, or maybe at cycle 32. وبدك تعطيه كمان cycles عشان توصلها. إذا كان عندك 100 copies of DNA of template راح تبلش من 100 أول cycle, ثاني cycle 200, ثالث cycle 400, رابع cycle 800, بعدين 1600. So you have a lot more. So each more كل ما زادت concentration كل ما زاد إيش الfluorescence. So you will have fluorescence at an earlier level. That's what this is quantification. So you can quantify the DNA this way. qPCR measurements. So Um, هاي السلايد شو فيها really nothing that we just added here, which is اللي هو تبلش من 1 تصير 2 4 6 8 whatever, or but different than 100 200, which is basically starting template. هنا this is it. One maybe a small thing is and see this is comparison between a sample with an unknown amount of template DNA and a standard. You can use a standard to give you an idea what is a high and low concentration, and then you can run it alongside your template, your template that you're looking for, so you can have an idea to compare it with. Basically, that is something that can be used. This is the exponential phase, linear phase, plateau phase. So here growth is being exponential, and then you go start linear once you have start making, and then this is the plateau phase that we just mentioned. Nothing really here that we did not mention. So by using multiple dilutions of known amount of standard, this is you can generate a standard curve. The amount of DNA or cDNA in an unknown sample can be calculated from the CQ value, and we can use multiple primers for different sequences. تمام. هلا اللي احنا بنساويه in real-time PCR is this fluorescent dye based method. So you have a double strand. لو رجعنا صورة عنها بتشبهها. You have a double stranded DNA template. This is the ground state fluorophore, and this is the excited state fluorophore. When it's double stranded, it's excited, denatured, annealed extension, to resulting strands, and then double the amount of fluorescence. هذه one method. There's another method for real-time PCR where you use a probe. A probe would be complementary to a specific gene. So إيش معناته? This probe will only work if the template has the gene that you're looking for. If the template does not have the gene, this probe will not anneal. If it annealed, if the sequence is there, it will anneal. Once the polymerase comes, it will break off the fluorophore, break it off. Once it's broken, it will become in the excited state. If it is excited, it is fluorescent. So the more probe the binds, that gets incorporated into the double strand, the more probe is being removed by the polymerase, and the more excited state fluorophore that you have concentrated بزيد الconcentration تبعهم, and you get the same thing. So you still have background level, and then the threshold level, and then the same thing, but this is just a slightly more specific way of doing it. I'm leaving this for you just to look at different methods for gene translocation detection. مثلا رح نحكي عن فيش محاضرة قادمة. هون this is an RT-PCR fusion transcripts of this liposarcoma, and then real-time PCR amplification with several samples. طلعنا هلا this method. آخر شيء نحكي عنه هو agarose gel electrophoresis and DNA staining. This is something that you will not do in a qPCR. كل هذا ما راح تسويه بالPCR. أنت بتسويه بستاندرد PCR. So in a standard PCR, you don't have the curve. You don't have a computer screen that gives you هذا بعطيك curve. You have to go get the DNA. بعد ما تخلصها بتاخذ الرياكشن بتنظفه. There's there's cleaning for the PCR. Sometimes you can use the PCR right away. بتحط عليه dye عشان تعرف وين حالك بالجل, and then you load your DNA into these wells. زي هيك, these wells. So wells that you add إذا معكم شفتوها. So you add the DNA after you dye it. This is a negatively charged stain that can move alongside. It's just a dye that tells you وين, لأنك DNA أنت ما بتشوفها بس عشان تعرف حالك وين DNA وصلت. So you put these in the wells, and then you run هاي tray, and then you put it in a casting tray. بتحط السامبلز تبعينك. So in a buffer solution, and then you run. This is the voltage source. بعطيك voltage. مشي لك من the negative to the positive, and then because DNA is negatively charged, it will go towards the positively charged, and then بضلها ماشي. شو بصير وين كنا؟ تمام. So you start running from the negative end towards the positive end, and then the bands will migrate. الباندز جوا الدي ان اي they will migrate. كل ما كبر الدي ان اي كل ما كانت slower in its movement. كل ما كان دي ان اي الموجودة اللي انت بالبي سي ار تبعتك صغيرة كل ما مشيت اسرع. So you put this called a marker. هذا marker فياته انواع احجام مختلفة من الدي ان اي 1000 2000 900 800 600 انواع مختلفة. So you can compare it to your samples. this is you put your sample and you run the current. this sample مثلا حجمها 1000، هاي حجمها 800 مثلا whatever it is. هاي حجمها 600، هاي حجمها 300، هاي 205، هاي 100. So that's how you look at it. Can you look at the DNA right away? That this is where ethidium bromide staining comes. Ethidium bromide يا جماعة بشوفكم تشتغلوا أحيانا. It is can bind DNA. It is carcinogenic. It is highly carcinogenic. The way that it works, it binds DNA, and then it is excited by UV light. So that's how you can visualize. هون you basically هاي الدي DNA ماشي، هاي الدي DNA الباند تبعك. With UV light you can see it. So you can then in a special camera box you can take a picture of this, and then you will come up with this. ترجع بيجيك هذا الجل image, and then you can or this or whatever, and then you can come up and then you can analyze your gel. So it's very important. That's how gel electrophoresis works. كتير مهم. محاضرة ما خلصت هلا، بس أنا راح أخلص حكي. بدي تفتح لي على هذا الفيديو. This is about gel electrophoresis. This is very important. This is good, and then it gives you about an idea about blotting. المحاضرة الجاي PCR and how to troubleshoot PCR. This is a good video, and then about qPCR. Watch these videos. I think I covered everything in these videos, but it's good to watch it again just to keep it as your reference. ترجع لهم متى ما بدك. And ويعطيكم العافية إن شاء الله. This is where we stop.